In Chapter 3, we're going to talk about how the genome in various organisms is structured, organized, and where variation comes from in particular genomes, whether that is between different species or between different individuals within the same species. I will apologize, this is a large chapter, it's one of our bigger ones, so the lectures will be a little bit long for this one. And I always struggle with this chapter because I want to cut things out, but when I get down to the things that I think I'm going to cut out, I realize that we need the information later. So generally I try to keep the lectures under 2 hours total, or roughly 2 hours total. This one is probably going to go over just a little bit, and I apologize for that. That is not the norm, but I could not cut any more out of this chapter without taking out information that we desperately need for foundation. So seeing as this is early and the whole point of our genetics class is to learn about the genome and how it functions and how it's inherited, I didn't cut too much out of this one. There are some things in here that trip students up, some vocabulary, things like that. So just make sure that you're keeping track of and organizing your notes. Make some flashcards here. There's not a lot of vocabulary, but a lot of different concepts that we address here. Before we get started, I wanted to do a quick review from some information in Chapter 2. So here's a sequence of DNA, just a short sequence. And just by way of review, remember that DNA has a direction, and we say that it works in a 5' to 3' direction. Now the significance of this is that a new nucleotide would be added here at the 3' end if we needed to do that. Remember that DNA is double-stranded, so let's just draw the straight line to indicate the other strand, and complementary. We'll talk about that in just a second. It's also antiparallel, which means if this 5' end is here, the other side is going to be the 3' end of the antiparallel strand. So those notations are important when you're writing DNA strands. Make sure you always label the 5' and 3' end. So if we have this sequence of DNA, what is the other strand, the complementary other side of the DNA strand? So we're going to start out, we use this template or this DNA as a template. The complementary strand will run 3', and then remember that G always pairs with C and T always pairs with A. So we can continue making our strand here using complementary nucleotides. Hopefully I don't make any mistakes here, but always make sure that you're paying attention and hopefully let me know if I make any mistakes. And then of course the other end is going to be 5' on that end. So if we were going to add a nucleotide, if we were doing replication or transcription, we would add a new nucleotide to this end here. So we have our complementary strand here. Based on this sequence here, is it possible to determine if this sequence is template or coding? Why or why not? So in this case, we can't actually tell if it is template or coding. We're just given a DNA sequence and told to make the complement. But remember, when you are transcribing something, knowing which is the template and which is the coding strand is really important, because your template strand is the one that you're going to use to make the correct sequence that mirrors your coding strand. So let's say now that this is a template strand of DNA. Let's figure out what the messenger RNA would be based on this template. So our messenger RNA is also going to run antiparallel to our DNA sequence. So we'll have a 5' end here, 3' end here. Remember that messenger RNA is going to be made as a complement to the template, and we're going to insert a uracil every time we would need to insert a thymine. This is because RNA utilizes uracil instead of thymine. So the complement for the T is an A, and we're adding in this direction simply to demonstrate the 5' to 3' addition of nucleotides and the anti-parallel direction. So when we have a G, we insert a C, C insert a G, T insert an A. When we get to the A, if it was DNA, we would insert a thymine, but because it's mRNA, we're going to insert a uracil. And then we'll just continue on for the rest of our sequence. So now we have our messenger RNA. So now what is our coding sequence? Well, our coding sequence is going to be complementary to the template. It's also going to be the same as our messenger RNA, except that we'll put T back in instead of uracil. So we can use either the template or the messenger RNA to determine what our original coding sequence was. So here, because messenger RNA and coding DNA are essentially the same, we're not going to go anti-parallel. We're simply rewriting. And when I say not anti-parallel, I mean with respect to that messenger RNA. We are going to be anti-parallel with respect to the template. So in our coding strand, we'll be using T instead of U. Otherwise, everything is the same as the messenger RNA. Hopefully I got all those correct. All right, so you can see that the messenger RNA and the coding strand are the same with the exception of the uracil and thymine switch. The template DNA is complementary to both the messenger RNA and the coding RNA. All right, let's look at one more thing by way of review from chapter 2. So remember that our coding molecule is always bound by complementary base pairs. So that means that A always binds with T and C always binds with G. Now remember, this is one purine and one pyrimidine each time. Adenine binds with thymine by 2 hydrogen bonds, and cytosine binds with guanine by 3 hydrogen bonds. So this complementary base pairing rule was developed by someone named Erwin Chargaff. And Chargaff was studying the composition of DNA molecules. Now, he worked extensively trying to determine what components make up DNA, and throughout his experiments, he made 3 generalizations based on what he learned. Today, these generalizations are known as Chargaffs Rule. So, Chargaffs Rule says the number of adenine always is equal to the number of thymine bases. That's because they're complementary. Likewise, cytosine is always equal to guanine. Now, the other thing that we can learn from this is the proportion of adenine and thymine is the same, and the proportion of cytosine to guanine is the same for both strands. That makes sense because, again, they're complementary. And then finally, Chargaff noticed that each species has its own ratio of adenine to thymine and cytosine to guanine, and that ratio is fixed within a particular species. So all yeast organisms have the same, or a specific species of yeast has the same percent of adenine as thymine, and the remaining percent is made up by equal portions of guanine and cytosine. So we can use this information to tell us a little bit about the molecular composition of a DNA molecule. So let's look at yeast first. If 30% of our bases in a DNA molecule, 32.9% of the bases in a DNA molecule is thymine, then 32.9% should also be thymine. Now, the remaining 40-ish% or so is going to be made up of guanine and cytosine. So if 17% of the genome is cytosine, then 17% of the genome is going to be guanine. Likewise, we can do the opposite with the sea urchin. If we know that 32.8% of our genome is adenine, then 32.8% should be thymine. Same thing with our guanine-cytosine ratios. Now, in the actual experiments, he did not get necessarily the exact same ratios every time. In his real experiments, for example, with the yeast, he actually got 31.3%, 32.9%, 18.7, and 17.1. Now, this just has to do with some experimental error and things like that. Don't get too bent out of shape worried about it. On an exam, I would give you a number and you would give me the identical number, assuming that adenine always equals thymine, for example. So don't worry about that too much. My numbers here aren't going to add up exactly to 100 because I've rounded and estimated and whatnot. But we can still use this information to determine what's going on in other organisms. So if we look at herring, which is a fish, and 22.6% of our genome is cytosine, that means that 22.6% is going to be guanine. Now, if we calculate the remaining percentages, so that is, I'd say, roughly 50%. If you actually calculate it out, you're going to get roughly 27.5% thymine and 27, I know that's not exact, 27.8% adenine. So I'm sort of moving back and forth between estimating and actually using Chargaffs Rule experimental numbers. Just keep in mind that your adenine is always going to equal or roughly equal thymine. Cytosine is always going to roughly equal guanine. So then for humans, if we have 31.2% thymine, then we're going to have roughly 31.2% adenine. Chargaff's actual percentage was 30.7. And then same thing with guanine and cytosine. You could calculate that out, decide what's remaining. He actually measured 19.3 and 18.8 for his percentages of G and C. But overall, you can see that they are roughly equal. We have roughly equal percentages of adenine, thymine here, roughly equal percentages of cytosine and guanine. And these remain constant per species. So let's move into chapter 3. As I said, chapter 3 is a huge chapter. We are going to talk about how genomes are structured, how they are organized, and how changes occur. And then a little bit about how that actually affects the phenotype of an organism. So let's look at just sort of a brief overview of our chapter. And it's important to keep in mind that evolution impacts the structure and the variation within a genome. If you need a review, chapter 1 has an excellent review on evolution. It might be good if you haven't read it already to at least skim it and just sort of to remind yourself what's going on in evolutionary theory. Second, variation is required for evolution. So evolution affects the structure of the genome, it affects the variation of the genome, but evolution is only effective if there is actually variation in heritable traits. That means that individuals have to have some differences in their genomes in order for evolution to occur. So, stepping back a little bit, we already know that genomes encode information that directs biological processes within an organism. That's the whole point of DNA acting as a genetic code book. And then, of course, we have differences in genomes between organisms. So this is how organisms, or why organisms, are different. They have different sets of instructions, and those instructions have been molded and formed based on the processes of evolution. So different species obviously have different DNA. They have different sequences and they have different types of organization. Genetic variation is a product of natural selection, so the environment affects what genes are present in a particular population and in a particular organism, and it affects how well those organisms are adapted to their particular environment. So those same processes also affect the entire genome. So genome organization, the number of genes, the types of genes, all that sort of thing are affected by natural selection and evolutionary process. And therefore, by extension, the process that affects the genomes affects the species as a whole. Remember, Charles Darwin is sort of the founder of evolutionary theory. But there was another man named Alfred Russel Wallace who did very similar work to Charles Darwin, but he was working in the Malay Archipelago, which is in the all those islands there off the coast of Indonesia. And he found very similar data, very similar observations to Charles Darwin. He just didn't publish it before Charles Darwin did. They knew of each other, they corresponded, but Charles Darwin published his information first. All right, so natural selection is an important mechanism of evolutionary change. So when there is variation in traits, you get differential reproduction. This means that some individuals are more likely to survive because of those traits. So that is natural selection and adaptation. That means that, this leads to differential reproduction. That means when those individuals are more likely to survive, they are also more likely to reproduce and therefore pass on their genes. So when we have variation and differential survival of a hereditary trait, we get evolution by natural selection. So in order for evolution to occur, we need all of these things. We need variation in traits, we need differential reproduction and survival, and we need those traits to be heritable. So when evolution occurs, it is based on the selection strength of a particular genotype. So as evolution is affecting the genome, we need to look at the strength of selection. So a trait will increase or decrease in frequency depending on how it is being acted upon. So let's take a look at what I mean by this. If we have a particular trait, and remember this trait has variation, it means there are different versions of this trait, so maybe this is 2 different fur colors or something like that. Differential survival, and is heritable. So this trait is encoded in a genome. So there is a genotype associated with this trait. Now, if we have positive selection means that the trait increases survival. That means the trait is going to increase also. So the frequency of the trait increases. If we have very strong selection, that means that individuals have a very large advantage. That trait's frequency is going to increase in the population very rapidly. Now let's say that we have a better chance of survival with a particular trait, but it's just a slight advantage. That just means that the trait won't increase as rapidly. So the trait increases in the population more slowly. We can also have negative selection. That means individuals with a particular trait have a decreased chance of survival. that means that individuals with the trait are going to decrease, and therefore the trait is going to decrease in the population. Now we can have the same pattern if it is very disadvantageous to have that trait. If it's very detrimental, that trait frequency is going to decrease rapidly. So the trait frequency decreases rapidly. And let's say it's not so great for the individual, but not quite as bad. The trait's going to decrease more slowly in the population. So by extension, when we have strong pressures on a genome or strong pressures on a phenotype, and therefore a genotype, we get rapid changes in the genome. And then, of course, we could have lesser pressures that affect the phenotype, therefore the genotype, and then slower changes in the genome. But keep in mind that evolution and genotype are very tightly linked. So if you have evolutionary forces causing a change in a phenotype, you're also going to have changes in the genotype. When some individuals survive and reproduce better than others, we say that they have higher fitness. Fitness differences can result from many things, but they all have to relate back to an underlying DNA sequence if evolution is going to work on them. So if one individual is going to survive better than another, it must pass on that trait in order for that trait to accumulate in a population. So how does that selection actually relate to underlying DNA? Well, selective differences can arise based on a variety of things. So keep in mind that selection is working on a particular phenotype, and that phenotype has a corresponding genotype. Now that genotype is going to relate to differences in DNA sequences. And they can differ between, let's say, a normal and a mutant, or something like that, mutant phenotype. Differences can arise based on timing of when and where transcription occurs. So we talked about that very briefly when we in chapter 2. This is due to transcription factors. So we can have differences in expression due to regulation by transcription factors. So if you have two organisms with different transcription factors working, then selection could possibly work if one of those is expressing genes in a particular time or place that's more advantageous. That gene is going to increase in the population. Likewise, if you have transcription factors that are affecting the amount of transcript made, so maybe a gene is being expressed more often, selection can work on that, a gene being produced at a different rate. You can also have altered amino acid sequences. So this is actually a change in the protein, but remember that protein is coded in the messenger RNA and therefore the DNA. So if you have changes in amino acid sequence, those are going to be reflected in the DNA as well. Now you can also have selection working on proteins themselves. So maybe in one individual you have a genotype that corresponds to a particular protein interaction or combination of protein interactions, and those compared to another individual that has coding for a different type of interaction. So there are many different molecular mechanisms that selection can be working on. And then, of course, you can simply have a mutation or a change in DNA. So if you have a different genotype for a particular gene in two different individuals, you are going to get a phenotype that differs. And really, those mutations can affect any of these differences in expression. So we know that genetic variation is needed for natural selection, but how do we, as scientists, actually see that variation in the genome? We might look at two individuals and see that they have a different phenotype. But in a laboratory, we want to get more specific. We want to see where that underlying genetic or genotype variation is. There's a couple of ways we can identify natural variation in a sequence. So we can do this by looking at proteins. So we can perform polyacrylamide gel electrophoresis. So this is very similar to the electrophoresis that we discuss in one of our labs, electrophoresis of bacteriophage DNA. Remember that in electrophoresis, you have electrodes that are positively and negatively charged, and when you load the DNA, the negatively charged DNA is going to run towards the positively charged DNA and spread out. The same idea applies to proteins. Proteins are also charged, and so you can run them on a gel in a similar fashion. The minor difference is that the gel is vertical and you load it from the top down and they run down through the gel. But they are still the chamber is still charged, and the size and charge of the protein still produces bands of protein. And remember, if we know about protein differences, we can infer DNA differences. We can also look at changes in DNA sequences using something called restriction endonucleases. So restriction endonucleases, I'm going to abbreviate them RE for short, are basically a bacterial defense mechanism. And this defense mechanism has been utilized in laboratory experiments. We'll get back to restriction endonucleases a little bit later in the semester, but just as a quick introduction, if you have a double-stranded piece of DNA, you can use a special protein to bind to that DNA, and that protein is going to cut your DNA at very specific locations. And so you end up with fragments of DNA. Let's say this same restriction endonuclease also makes a cut here. You would end up with 3 fragments. Now, the same DNA, so say my DNA, is always going to cut with the same pattern. But if there are differences in a genome, say between 2 people, you're going to have differences in the pattern. So maybe this is my DNA, and I get only 2 fragments, but maybe this is my brother's DNA, and he has 3 fragments because he has a change, the sequence change here. So these differences are reflected in something called RFLPs, restriction fragment length. So we're going to look at the different lengths of our fragments, polymorphisms. So poly means many, morph means shape. So you can get different patterns depending on these restriction enzyme sites. So let's look at an example over here. Let's say that this restriction enzyme known as EcoR1 cuts at a very specific sequence. All restriction enzymes cut at a specific sequence. And so if EcoR1 makes a cut here, we are going to get 2 pieces of DNA. And there's our DNA, our 2 pieces on our gel. Let's say that another individual has a change at this location here. And so that EcoR1 restriction endonuclease is not going to cut at that site. And so this individual gets only 1 band. So now these pieces, the restriction fragments, are different lengths. So we can see the different lengths reflected in the polymorphisms in the genome, or the changes in the genome. We can also identify variation based on DNA sequencing. So over the past few years, DNA sequencing has changed rapidly. The first type of sequencing was called dideoxy sequencing, and I'm going to talk about that very briefly, just the general idea behind it. More commonly now, we do something called massively parallel sequencing. This is just a quicker way to sequence many types of genomes at the same time, but the general underlying mechanism for sequencing is the same. The differences have to do with how the actual experiments go and how the computer analysis goes. The importance of DNA sequencing is that now we have complete sequences of many genomes, and genomes are becoming more and more available. This allows people to quickly find genes, quickly determine functions of genes. So if you study a gene in one organism and you find it in their genome, and you sequence it, it's probably going to work similarly in another organism based on evolutionary history and relatedness of those organisms. So being able to have a library of DNA sequences is really important. So understanding and having those sequences is really important in determining functions now, past functions, how functions have changed over time. Basically looking at sequence evolution along with organismal evolution. So before we can talk about how DNA sequencing works, I want to introduce something called polymerase chain reaction, or PCR. We'll do this in more detail when we talk about replication, and we'll do it even more detail when we actually do it in lab. But polymerase chain reaction is basically DNA replication in a bottle. So you need a few things for this. You need a DNA sample. You need primers, which are oligonucleotides. In chapter 2, we talked about short sequences that you can bind or hybridize to a DNA sequence. And those are going to bind to our DNA and essentially start the replication process. They're like little tags to isolate the region that we want. And then we need our nucleotides because we're going to be replicating DNA. We need all the pieces. We need something called Taq polymerase. Now this is an enzyme that is actually going to perform the addition of nucleotides. So it's actually it's an enzyme that adds your nucleotides to the growing strand. And then we have some reaction solution, and then we mix it all in a little tube. So the idea behind PCR is that in the first step we denature. This means we pull apart our strands, and we do this by heat. Then we add our primers, our oligonucleotides. And what those are going to do is just isolate the section that we want to copy or sequence at a particular time. So then we allow them to anneal. That means to come together. We bring in our oligonucleotide, our oligos anneal, or they bind to our fragment. Then Taq polymerase comes in, and we'll talk about this more in the next chapter when we talk about replication, but Taq polymerase comes in, binds to the strand, and then copies it. It adds those DNTPs in the extension phase, and what we end up with are these two newly copied strands. These outside ones were the template that was pulled apart, and inside we have the new strands. Now, PCR is really cool because every time you do this, you double your number of fragments. So every time we go through a cycle, we do the denaturing, annealing, extension phase, and every time we double what we have. Now, this has a lot of different uses that we'll talk about later. You can use this for all sorts of different analysis with your DNA. Right now, we're going to focus on how this relates to sequencing. So in sequencing, you have a template, and you're going to bind a very short primer. I forgot to say, this is the sort of first widely used method of sequencing called Sanger sequencing. Excuse me. And this is the method that the Human Genome Project used in the 90s. Now, it's time consuming and it takes a lot of people doing it, a lot of computer analysis. It still takes a lot of computer analysis, even though we've streamlined the process. But essentially what happens is you perform PCR in many, many different steps. So you start adding your nucleotides, and you have a mixture of regular nucleotides and those that are tagged with a special signal. So these are tagged, and occasionally a tagged one gets incorporated. And when it is tagged and gets incorporated, it stops the PCR reaction. And so if you run this long enough, you get a tag at every single base pair. So if we start out our sequence with CTG, and then we add an A and stop, there's our shortest fragment. That's going to be this little guy down here. Then if we run it long enough, we'll get a fragment with a T incorporated here. There's our next one. Then with our G incorporated here, there's our next one. And you run this through a gel, and the first one, because it's smallest, is actually going to run out the bottom of the gel, and a little sensor here is going to read this tagged nucleotide, and it's going to say, oh, first position A, second position T, and it just, as these run out the bottom, it actually reads what the last one is and gives you a sequence. So here we have our G, C, A, T, etc. This is not exactly the same sequence, but you get the idea. So these peaks are basically how many times they read it. So you can be pretty confident that these spaces, well, the higher they are, the more confidence you have in them. And so then you use computer software to analyze this sequence. Now this worked by using small fragments. They would sequence a fragment at a time, or a long fragment, and then if there were smaller sequences, they would be compiled sort of in overlapping, called contigs, overlapping sections, and those sections would be analyzed for computer, and you would get a readout based on the sequence that you got from your gel. So newer sequencing methods break the DNA sequence up into smaller pieces, and this is much faster. So you can essentially do more DNA in a shorter amount of time. And by using smaller fragments and lining them up, you get multiple sequence events. And so you can be more sure of your actual sequence. Some sequencing is quite cheap. There are different types of sequencing. If you go back to the toolbox, I believe it's 3.2 in your textbook, it actually talks about some of the different very specific ways that we have new different types of DNA sequencing. Some methods allow you to actually sequence RNA instead of making a cDNA library, and this is pretty cool because until recently you couldn't really sequence RNA because the methods were not fast enough before it broke down.